FSc Biology Quick Summaries, Chapter 20, First Half, Chromosomes and DNA for the preparation of PPSC Lecturer and Subject Specialist Zoology and Biology. In this first half of Chapter 20, we shall discuss chromosomes, types of chromosomes, and moving through all these topics, we shall end at Meselson Stahl experiment to prove whether the replication is semi-conservative or not. There came a person to Prophet Sallallahu Alaihi Wasallam from Banu Fazra and said, "My wife has given birth to a child who is black." Whereupon Allah's apostle said, "Have you any camels?" He said, "Yes." He again said, "What is this color?" He said, "They are red." He said, "Is there a dusky one among them?" He said, "Yes." "How has it come about?" He said, "It is perhaps the strain to which it has reverted." Whereupon the Prophet ﷺ said, "It is perhaps the strain to which he has reverted." Sahih Muslim Kitab al-Layan, Hadith number 1500. Let's start. Chromosomes Introduction. Chromosomes appear in nucleus during cell division only, and all the rest of the time they are in the form of chromatin threads and they are invisible. They were first discovered by Walter Fleming in 1882 in salamander larvae when he was studying that larvae. Penicillium has only one pair of chromosomes. One pair means it has two chromosomes. Mosquito has 6, corn has 20, frog has 26, honeybee 32, mouse 40, and humans have 46 chromosome, 23 pair. Now you may be asked in the exam that how many chromosome pair corn has, so that will be 10. So please remember total number of chromosome as well as their pairs. Sugarcane has 80 chromosomes and some ferns has up to 500 pairs of chromosomes. Please remember. Types of chromosomes. Chromosomes have chromatids, centromere, primary and secondary constructions. Chromosomes differ in size, staining properties, centromeres, arm length and constrictions along the arm. So these are the features which differentiate one chromosome from the others. The particular array or arrangement or look of or appearance of or morphology of chromosomes that an individual possess is its karyotype, and that karyotype of the same species almost same but it is different in different species. Usual shapes of chromosomes are I, J and V. Now telocentric chromosomes has one long arm and centromere at one end or terminal, and they are I-shaped, and telocentric chromosomes are not present in humans, while acrocentric has one very small arm and other large arm with centromere at one side. Now the major difference between acrocentric and submetacentric is that the small arm in submetacentric is relatively large than acrocentric, but it is J-shaped and acrocentric is I-shaped. Metacentric has almost equal arms with the centromere at center and is V-shaped. Please remember these types of chromosomes on the basis of their size and position of centromere are very important for PPSC. Composition of chromosomes. Chemically, chromosomes have 40% DNA and 60% proteins. RNA is also associated with chromosomes because it is formed in the DNA during the process of transcription. So whenever a DNA is chemically analyzed, RNA is also found. Typical human chromosome contain 140 million nucleotides, that is of one chromosome, typical chromosome as exemplary chromosome. This amount of information will fill 280 books with 1000 pages, and if strand of DNA is laid open from one chromosome only, it will be 5 centimeters long. After every 200 nucleotides, DNA is coiled around 8 histone proteins to form nucleosomes. So nucleosome is the coiled around DNA histones. Histones are positively charged due to abundance of arginine and lysine. Arginine is a positive amino acid. DNA is negatively charged due to phosphate groups. Highly condensed portion of chromatin is called heterochromatin, and some portion of heterochromatin remain permanently coiled without being expressed ever, that may be consisted of introns. Euchromatin condenses only during cell division, and rest of the time it remains open or uncoiled. Chromosomal theory of inheritance. Mendel's work was rediscovered by German geneticist Karl Korens. Walter Sutton first formulated the chromosomal theory of inheritance in 1902. In 1910, T.H. Morgan detected mutant male fly having white eyes instead of red. Now please remember, this fact is very important with respect to PPSC. The mutant white-eyed was a male fly, not a female. He crossed mutant male with normal female, and all the progeny and offsprings had red eyes, and the white color was hidden or masked. Then he crossed F1 with each other, and he found 18%, almost 782 offsprings having white eyes, and in contrast to Mendel's theory, all white-eyed were males in that F1 generation. Then he crossed white-eyed male with F1 red and obtained both white-eyed male and female. Why this happened? The reason that gene causing white eye is recessive and present only on X chromosome, not on Y chromosome. So a female have 2 X chromosome, and if that recessive gene is present on one X and the other has dominant, then that will be a carrier and no white color will be shown in the female. That was the reason in F1 all the white-eyed were males. Eye color assort independently because chromosomes do assort independently, hence proving Mendel. We shall discuss all these crosses in the upcoming chapter in detail, that how these heterozygous, homozygous individuals produce the offsprings or progeny. DNA as hereditary material. Frederick Griffith first provided evidence of hereditary nature of DNA. Please remember, Griffith. He infected mice with virulent Streptococcus pneumoniae bacteria, and mice died of blood poisoning. Now the blood poisoning is important here because no one focus on such type of factors. So how mice died due to blood poisoning? When he infected mice with mutant strain with no virulent polysaccharide coat, initially at start it was thought that the polysaccharide coat is actually responsible for the disease, and the mice showed no ill effects. Pathogenic form of Streptococcus pneumoniae is referred as S form because it forms smooth colonies on a culture dish in the laboratory. Non-virulent form which do not have enzyme to manufacture polysaccharide coat form rough colonies, so is referred as R form. So S form or R form are basically due to the nature of the colonies which they form on the petri dish in the lab. To check whether polysaccharide coat is pathogenic, Griffith injected dead S strain into mice, and mice remained healthy, so rejecting the hypothesis that the polysaccharide coat is responsible. As control, he injected mixture of dead S strain and live coatless R strain. R strain do not have coat. Unexpectedly, mice developed symptoms, and many of them died as a result of this injection. Why this happened? Blood of dead mice then was analyzed, contained live virulent S strain bacteria with surface proteins of previously R strain. So that kind of transformation has occurred from R into S form. The agent responsible for this transformation remained undiscovered till 1944. Avery MacLeod and McCarty referred this as transforming principle, and they first prepared mixture of dead S strain and live R strain bacteria. They removed proteins from mixture up to 99.98%, that almost there was no protein, but transformation still occurred. That kind of observation suggested that proteins are actually not the transforming principle. Now, protein digesting enzymes, RNA digesting enzymes placed in that medium could not stop transformation, but when DNAs were put in the medium, they destroyed the DNA actually, and the transforming activity was stopped or inhibited. So they concluded that neither RNA nor proteins are the transforming principle, but actually the transforming principle is DNA. Alfred Hershey and Martha Chase provided additional evidence in 1952. They worked on bacteriophages T2. They labeled viruses with isotope P32, phosphorus 32, and which was incorporated in the DNA. In other experiment, they used isotope S35 for proteins. Sulfur is present in the proteins and phosphorus is present in the DNA. That's why they use the two different kinds of isotopes. These labeled viruses were allowed to infect bacteria. Bacteria quickly reacted to remove proteins from surface having S35 in them, but P32 entered bacteria along with DNA, and when new phages which were released from that bacterium after lysis all had P32 in their DNAs. This proved that hereditary material is DNA, not protein. So remember the name of the scientist, Alfred Hershey and Martha Chase. Chemical nature of DNA. German chemist Friedrich Miescher discovered DNA in 1869, just after 4 years of Mendel's work published. Now please remember that DNA was actually discovered by Friedrich Miescher. He called white substance extracted from human and fish sperm as nuclein. So Friedrich Miescher called that nuclein, and due to this nuclein, because this substance was acidic in nature, that's why it came to be nucleic acid. So actually the word of nucleic acids for DNA and RNA was derived from the word nuclein, and due to the acidic nature of that nuclein. Now Levine determined 3 components of DNA first of all, and these were phosphate group, pentose sugar, and nitrogenous bases, both purines and pyrimidines. He further concluded that DNA and RNA has repeating units of nucleotides. So all these works were done by P.A. Levine, please remember. Nitrogenous base attach with carbon 1 of sugar, while phosphate to carbon 5. Now 1 and 5 pair, please remember that for attachment. Free hydroxyl group is attached at carbon 3. So 5 phosphate and 3 hydroxyl group allow DNA and RNA to form long chains. Please remember, these are very, very important facts. The reaction between phosphate of one nucleotide and hydroxyl of other is a dehydration synthesis by eliminating water molecule to form covalent bond, and that is the basis for the elongation of the polynucleotide chain. So phosphodiester bond is a covalent bond, and it is called diester because phosphate group is linked to 2 sugars by a pair of ester bonds on either side. Every strand of DNA or RNA has one free 5' phosphate group on one side and free 3' hydroxyl group on other side, and these 2 terminals can be used to elongate that strand further. Erwin Chargaff showed that amount of guanine equals cytosine and adenine equals thymine in the DNA, and that was a very remarkable discovery, and many of the questions in the past PPSC have been asked regarding Erwin Chargaff. Rosalind Franklin carried X-ray diffraction analysis of DNA, while Maurice Wilkin or Maurice Wilkin prepared DNA fibers for analysis. Now, these two were working together. X-ray diffraction techniques were employed by Rosalind Franklin, while the DNA sample was prepared by Maurice Wilkin. They found that diameter of DNA is 2 nanometer, while the each turn is 3.4 nanometer apart. Watson and Crick's model of DNA. Base pairs in a DNA are always pointing toward each other. Purines are larger bases, while pyrimidines are smaller ones. Base pairs are planar and stack 0.34 nanometer apart as a result of hyperphobic interactions. Now, these hyperphobic interactions actually contribute to stability of the molecule. They suggested that basis for copying the DNA is complementarity. DNA replication is semi-conservative. So first Watson and Crick suggested the semi-conservative replication of DNA. In semi-conservative replication, primary structure is conserved primary structure while secondary is disrupted. Please remember, these are very important points. Conservative model suggests copying of new strands without opening older ones. Disruptive replication suggests the disruption of parental DNA into segments and then rejoining the previously older segments and new segments together. That is the disruptive replication. Meselson and Stahl. All the three models of DNA replication, that is a conservative model, disruptive model and semi-conservative model, were evaluated by Meselson and Stahl in 1958. They grew bacteria in a heavy isotope of nitrogen 15 so that it incorporated into DNA, and normal nitrogen is nitrogen 14. DNA with N15 was denser than DNA with N14. Now, they transferred bacteria from N15 pure medium, which all the DNA contained only nitrogen 15, to nitrogen 14 medium, and that is the key point here to understand the whole experiment of Meselson-Stahl. They first prepared all the DNA having N15, pure N15 DNA, and then they transferred that into N14 medium, that the medium containing nitrogen 14, not nitrogen 15. They collected samples from mixture at various intervals when the replication started. They dissolved DNA in cesium chloride and spun it at high speed in ultracentrifuge. Centrifuge is a machine, you know, for the spinning of the different solutions to separate them on the basis of density. DNA with N15 migrated deeper towards sodium chloride medium due to high density in the centrifuge. They concluded that after first round of replication in the mixture, now please remember, they have transferred N15 DNA to N14 medium, and if new strand will be constructed, that new strand will have only nitrogen 14 because N15 is already part of the strands, and new strands can only formed if they absorb nitrogen from the surrounding, which is N14. Now, after first round of replication mixture, hybrid strands were formed. So, hybrid strand mean the one strand of newly formed DNA was of N15 and the second was of N14. That suggests that the semi-conservative replication is the answer to the question. After second round, heavy strand produced hybrid, but lighter N14 strand produced both N14 strands or lighter ones. Then they proved semi-conservative replication of DNA. Next lecture, inshallah, chapter 20, second half, chromosomes and DNA. To watch other chapters, please check the playlist or description. Thank you so much for staying with me. Allah Hafiz.